Hi learners, I am Professor S. Kripanidhi, heading the Department of Biotechnology at Vignan's Foundation for Science, Technology and Research, a deemed university situated in a rural environment, Vadlamudi, Andhra Pradesh, India, to give a series of talks on immunology and immunoinformatics. Hitherto, we have presented 6 topics. The 7th topic now is entitled on antigen, hepten, and adjuvant factors affecting antigenicity. All these 7 topics come under the unit 1 of immunoinformatics. Okay, let us see now the antigen. Antigen is the one which is antibody generator. What is antibody generator? It is immune missiles. Antibodies are immune missiles to attack these antigens. Where are these antigens located? They're all located on the pathogens. Where are the pathogens? Pathogens are around us. We are not free from pathogens. We are living in an environment where pathogens are present, flora are present, fauna is present. We are surviving. So that's what now the government says, keep yourself alone. Don't make others infected. Don't make yourself to get infected. That's what we have to be away from this type of the pathogens and pathogenic antigens. Now, heptanes are the small molecules, the details of which we are going to see. Then another aspect here is the adjuvant. The adjuvant is the one which boosts immune response. It's a macro booster immune response. It is for experimental purposes. And again, factors affecting antigenicity also we see. A few of the pathogens are more virulent. A few of the pathogens are not virulent. If you are immunized, the pathogen is not virulent in us. We prevent the virulence. So that's what these are all the topics and that we are going to see in the 7th topic of Unit 1. So the objective is to familiarize with the complexity and nature of antigens, to know the definitions of various terms involved in dealing with antigen, immunogen, adjuvant, and heptanes, and to evaluate the factors affecting the antigenicity. All foreign materials are not antigens. If you have the attribute of an antigen, or immunogen, or a tolerogen, so those details we see now. The expected outcomes out of this topic is, 7th topic, the 2 terms relating to the antigen, namely antigenicity and immunogenicity will be known. Though both are synonymous, there is the variation in the involvement of the cellular components, antigenicity and immunogenicity. And now the response of the T cells in relation to the antigen complexity will be known. See, here we are using the other jargon called complexity along with the antigen. Okay, so the complexity make the T cell to get involved. Like this, the role of heptane. Heptane in eliciting immune response. Heptanes are the small molecules. They alone cannot elicit immune response. They require a carrier molecule. It is for the experimental purpose. That's what we are showing here. Immune potentiation through adjuvants will be appreciated. Whenever we take the vaccine is being coupled with adjuvants in order to improve the immune potentiation. So use of adjuvants in potentiation of immune response will be known. So let us see now.
Discuss 2 crucial but simplistic concepts in immunology. So I'll describe the difference between antigen and immunogen and the relation between them. So antigens are simply stated as substances which specifically bind to antibodies, in brackets the B cell receptor or the T cell receptor. So definitely these small proteinaceous or glycoproteinaceous molecules could bind to T cell receptor or B cell receptor, and that is the criteria they would be demarcated as antigens. Now these antigens could be found from specific pathogens such as bacteria and viruses, or itself inside the cell. The endogenous proteins could sometimes also serve as self antigens, and we would come to know that what is the function of these self antigens. Now body produce self antigens just to say just to convey this message to the immune system that our body is protected and our body is fine and don't hurt us. So it's like a protective flag. So there are surveillance cells such as NK cells. They always roam here and there and look for whether the cell is projecting the self antigens or not. So the self antigen gives a signal to disengage. So that is why self antigens are equally important as the antigens which are coming externally from pathogens. Now, there is another term which is known as immunogen. So as immunogen can bind to T cell receptor or B cell receptor, and not only they can bind to the T cell B cell receptor, they can also mount an immune response. So simply, the immunogens are kind of antigens which not only bind to the B cell and T cell receptor but also mount an immune response. So that is the difference between immunogen and antigen. So we can say that an immunogen is necessarily to be an antigen, but all antigens are not immunogen. Many antigens which are of small molecular size, known as hepten, they are not able to create an immune response by themselves, and they are going to only create an immune response when they are conjugated with several other carrier molecules. So immunogen is molecularly more complex than an antigen. So immunogenicity of any antigen depends upon its molecular size, complexity, and also the chemical composition and heterogeneity of them, and definitely its foreignness.
So just now the video that we have seen, very distinctly the difference between antigen and immunogen is shown. The antigen presenting through MSC molecule is shown. The macrophage presenting the antigen coupled with MSC molecule to T helper cell containing the T cell receptor complex is shown. T cell is interred, promoting the B cell is shown. So as a result, several of the components involved in adaptive immunity has been shown into this. But specifically, the distinct difference between antigen and immunogen is shown here. So now let us see. An antigen is a foreign substance which elicites immune response. Immunogen. The same definition goes by adding food word jargon called complex. It's a complex foreign substance which elicites T cell mediated immune response. That's what the immunogen. Now let us see, this is the antigen, it's a folded protein. The folded protein is having some ADS, antigenic determinant sites. Those antigenic determinant sites are called epitopes. In adaptive immunity, a memory B cell is formed means a memory B cell is formed for one epitope. A memory T cell is formed means a memory T cell is formed for against one epitope. That means an antigen may be having 50 to 100 epitopes. For each of this epitope, there is a memory cell formed in the adaptive immunity. Now let us see immune response to antigen. If an antigen encounters a naive B cell, effector B cell is formed. That's what we have been saying. All these immune cells to begin with, they are dormant. They are getting activated along the antigen presentation. So effector B cell is formed. Along the effector B cells, memory B cells form. And along the memory B cells, the plasma cell which gives the immunoglobulins. Like that, if you take up the antigen promoting this, encountering the naive T cells are going to become effector T cells and memory T cells. The memory T cells include T helper cells and T cytotoxic lymphocytes. So that's what the antigen activating, eliciting the immune response. What are immunogens? Proteins are the most potent immunogens because the proteins are highly folded, with polysaccharides ranking as second. In contrast, lipids and nucleic acids of an infectious agent generally do not serve as immunogens unless they are complexed with proteins. And one of the best examples of an immunogen is the hemocyanin. Hemocyanin is a copper-containing protein present in the arthropods and mollusks. It is having nearly more than 1 lakh Dalton's molecular weight, and it's having several epitopes, and hence it is considered as one of the best immunogen. Immunogenicity is determined in part by 4 properties. One is the foreignness. The second one is the molecular foreignness, which phylogenetically again Evolutionally, phylogenetically, taxonomically, it is far off from the host. And the second one is the molecular size. That is, if the antigen is around 5,000, 10,000, it doesn't cause immunogenicity. That's what what is it called? Insulin. Earlier, we used to have the insulin from bovine. We used to have insulin from pig. And insulin molecular weight is 5,000. They are not causing the antigenicity. Now we are having the insulin from the human. So that's what the molecular size plays an important role in determining the antigenic feature. Then chemical composition and complexity of the compound in particular of interest. Ability to be processed and presented with an MSC molecule. That is, whatever antigen that we take that is coming, it has to get anchored to MSC molecule. If it doesn't get anchored to the MSC molecule, then it cannot act as an antigen. Okay, so that one has to be presented by antigen presenting cell. That's what dendritic cell. Examples of foreignness. The greater the phylogenetic distance between the two species, the greater is the structural disparity between the antigens. For example, the common experimental antigen, bovine serum albumin, is the one which is being used in all biotechnology laboratories, is not immunogenic when injected into the cow, but is strongly immunogenic when it is injected into rabbit or mouse. So the molecular size. There is a correlation between the size of a macromolecule and its immunogenicity. Size of the macromolecule relies on the immunogenicity. The most active immunogens tend to have a molecular mass of 1 lakh daltons and above. Generally, substances with a molecular mass less than 5,000 and 10,000 daltons are poor antigenic immunogens. Complexity of immunogen. It is notable that all 4 levels of protein organization, that is, protein is having primary structure, secondary structure, tertiary structure, quaternary structure. They contribute to the structural complexity of the protein and hence affect the immunogenicity. Homopolymers, that is, single amino acid repeats, they are poor, they lack immunogenicity. Now let us see the protein structure, primary structure, where the assembly of amino acids takes place. Secondary structure, where is alpha helix and beta pleated sheet, where the folding is taking place. Tertiary structure, where packing is going on of these folded structures, and quaternary structure, where the interaction between these folded structures takes place. This is all the protein structure. Quaternary structure is highly immunogenic. Adjuvant, that's what about antigen. Now let us see the adjuvants. Adjuvants are substances that, when mixed with an antigen and injected along with the antigen, enhance the immunogenicity of that antigen. That's what whenever we take the vaccine, or human vaccines also, we are having inorganic adjuvants. That's what alum. So adjuvants are often used to boost the immune response when antigen has low immunogenicity or when only small amounts of antigens are available. So functions of the adjuvants are antigen persistence is prolonged. Okay, antigen persistence is prolonged. Otherwise, antigen is totalized. Co-stimulatory signals are enhanced. Local inflammation is increased. The non-specific proliferation of lymphocytes is stimulated. It facilitates for micro-booster exposure of antigen. This is an important aspect of an adjuvant. If you give an antigen as a vaccine along with the adjuvant, the vaccine contains hundreds and thousands of these antigenic molecules, and those antigenic molecules, one after the other, they make the host system to get exposed. That's what called the micro-booster exposure is being facilitated by the adjuvant. Types of adjuvants: alum, potassium, aluminum, potassium sulfate. Alum, it prolongs the persistence of antigen. It is being used for human applications. And there is another for experimental purposes, water and oil adjuvants also prolong persistence of antigen. A preparation known as Friend's incomplete adjuvant contains antigen in aqueous solution, mineral oil, and an emulsifying agent such as monopolyate, mannate monopolyate, which disperses the oil into the small droplets surrounding the antigen. That means antigen is circled by this type of adjuvant material. The antigen is then released very slowly. That means in a vaccine, we do not have one antigen molecule. Whenever any pathogen infects, we do not have one antigen molecule. We will have thousands of antigen molecules. So those thousands of antigen molecules along with the adjuvants are being encircled, entrapped in the adjuvant. They are gradually being released. So now, Friend's complete adjuvant. That we have seen complete. Friends incomplete adjuvant, this is Friends complete adjuvant developed by Jules Friend, contains heat-killed mycobacteria as an additional ingredient. Here, myramyl dipeptide, a component of the mycobacterial cell wall. This myramyl dipeptide activates macrophages. That's what macrophage has to be activated to engulf the antigen to produce the epitopes along with the MSC molecule, making Friends complete adjuvant far more potent than incomplete form. Activated macrophages are more phagocytic. Now we have seen the antigen, we have seen the adjuvant, now let us see the heptanes. Landsteiner employed various heptanes, that small organic molecules, to be antigenic but not immunogenic. So these heptanes are not antigenic on their own. They have to be coupled with the carrier protein. Carrier protein is non-immunogenic. Then antibodies will be developed. Heptane carrier conjugate. So along with the heptane carrier conjugate, we inject and we produce antibodies. The heptane determinant are produced and unaltered epitopes of the carrier protein, for which also the antigens are produced. New epitopes are formed, combining both heptane and carrier. So this is what the heptane. Heptanes are the small molecules. They are coupled with the carrier. As a result, now it has become a big molecule. So for this big molecule, the organism is, it is an experimental situation where the rabbit responds or mouse responds, the immune response comes. As a result, the immune response is elicitation of the immunoglobulins. The immunoglobulins against heptane, immunoglobulins against carrier, immunoglobulins against for both. So that's what heptane and carriers proposed with the Landsteiner, a renowned scientist. The summary aspect of it. So antigen, antibody generating substance elicites immune response, that is, it produces immunoglobulins. Immune response, elicitation of the immune response means immunoglobulins are produced. That's what the convalescent patient of the COVID-19 having the immunoglobulins. Those immunoglobulins are being used to treat other patients through the plasma therapy. That's what. A foreign protein usually presented by a pathogen called T-cell independent. Immunogen is the one which elicites T-cell immune response, where T-cell mediation is involved, that is T-cell dependent. There are super antigens, they are called, they require polyclonal T-cell response. Immunogenicity promotes cell-mediated immunity. Okay, heptane, small antigenic molecules require a carrier called heptane. Carrier heptane. And adjuvants are essential and accentuate immune response and used for the development of new and improved vaccines. Now the study questions for this topic 7. Distinguish between antigen and immunogen. What are epitopes? Elaborate the features of an immunogen. What is an adjuvant? Indicate its types. How does an adjuvant enhances immunogenicity? What is heptane? How does it elicit immune response? Give an example of anti-heptane antibody. Indicate the factors affecting antigenicity. With this, we completed the 7th topic of Unit 1, antigen, heptane, and adjuvant, and the factors affecting antigenicity. Please go through this. We'll be seeing the last topic, the 8th topic, in the next presentation. Thank you very much.